Hello and welcome to Biology Lesson 2.04, Polymers to Monomers and back again. In the last lesson, we talked about how we take something big like a piece of pizza and our body breaks it down, which is called catabolism, into monomers. And then our cells can use those monomers and build things back up. So what we're going to look at here is what is the reaction through which things get broken down and things get built up. So hydrolysis is we're going to see is the breakdown or catabolism. And we're going to see that dehydration synthesis is the building up or anabolism. So let's make these chains a little bit bigger. I know I have an OH group here, but I'm just going to put some more monomers on this nice big long chain. Because usually these polymers can be really, really, really big. So let's make it nice and big. And that means I need to add them here too. So I added 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So hopefully it's a little bit easy to see here that I have a great big long polymer. And let's pretend like my monomer is glucose. So here I have just one little monomer, and I want to attach it to this big long polymer. Now, if this is happening in a plant, we're going to see that the name of this polymer is starch. And you guys have heard of starch before. If you eat a lot of potatoes, it's called eating starchy foods. And that's because the name of the polymer in plants is starch. So we're going to build this up. We're going to connect another glucose to this big long starch chain. And remember, building up is anabolism. This happens through something called dehydration synthesis. If you've ever heard of the word synthesis before, you'll know that that means to build. So to synthesize something, it means to make or build. What's it mean to dehydrate? Have you guys ever felt dehydrated when it's a really hot day and you've been sweating a lot? Dehydrate just means to lose water. So when we go through dehydration synthesis, we're making or building by losing water. So my monomer of glucose here has some atoms that kind of stick off the side. And on one side I have that sticks off an OH. And I know in your head, if you didn't say it out loud, everybody just went IO. And on this side of my starch, I've got an H. The way that this gets added to this polymer is the OH and the H combine and then leave as H2O. In case you can't read it on yours, this is a water. This is a water molecule. When those combine and leave, it connects this to this, and we get a new bond. So we synthesized this new bond. I had a polymer that was 1, 2, 3, 4, 5, 6, 7, 8 long, and now I have a polymer that's 1, 2, 3, 4, 5, 6, 7, 8, 9. And I can keep doing this again and again and again. And in fact, this is what your body does. If you've been watching what you eat and then you've lost, you've lost a whole bunch of weight. You've lost 2 or 3 pounds, but then you sit down and you eat a big plate of spaghetti. Your body had broken down all of your carbs. And now it's going to take all that extra spaghetti the pasta that you don't use right away, the glucose out of that pasta, and it's going to start snapping these together through dehydration synthesis. You've ever made a pop bead necklace and you just pop the beads together. That's really kind of similar to what is going on, but we're doing it by losing a water. Well, what happens if you've had that pasta and the next day you're running cross country? So let's make our polymer a little bit longer again. I think this time I'll make them purple. Let's make this guy. How many did we have before? 6? So I have this big, long, and let's say this is a starch again. Let's say we've got another polymer of starch. But as I'm running cross country, I need lots of energy. I'm going to need lots of ATP. It turns out that's what we use glucose for, is to make ATP. So I can't put this into my mitochondria. It's too big. In the same way, if you have a wood burning stove at home, you don't cut down the tree and stick the end of the tree in the wood burning stove. You cut the tree into logs. Well, we don't have little tiny lumberjacks in our cells, so we have to break down these great big starch logs. Again, here's my monomer, just a red. So this is my glucose, which is a monomer, and my starch is my big polymer. So in order to get this guy free, I have to crack that bond. Well, we crack the bonds, we break the bonds through a process called hydrolysis. Hydro means water. And if you haven't had the word lyse yet on your vocab, you will soon. Lyse means to break. So we break bonds with water. We break bonds by adding water. Oops, I'm off screen. So the water comes in and the water gets pulled apart. Half of the water goes on to the glucose, the OH goes back to the glucose, and the H stays on the end of the starch. So again, the water comes in, the water molecule gets pulled apart, and now we have a free glucose, and we can send this guy to the mitochondria to get turned into ATP. Again, to build up anabolism, we're going to synthesize through dehydration. So what are our takeaways? We have anabolism, which is building up. We do that by dehydration synthesis, which is losing water and creating a bond. To do catabolism, we have to crack monomers off the end of a long polymer. We do that when water comes in, attaches, gets pulled into two, and breaks the bond through a process called hydrolysis. That's all we have today for 2.04. Thanks for watching and have a good day.